Alrighty, welcome back for the MCAT grind. This is Biology Chapter 4, Nervous System. Whereas pretty much MCAT's favorite question types and passage, they love testing about nervous systems, whether new type of drug that does this, does that, impact your nervous system, and will have you think back and know this in detail. In contrast, however, it would be a lot more application questions, so it's good to know the fundamentals here. Now, we have the neuron. It includes the cell body with the dendrites on the very tip as receptors with sites for neurotransmitters to bind to and transmit the signals down to the axon hillock, and it will send through saltatory conductions the electrical signals down the axon, and it will finally arrive at the nerve terminal. The information will then be passed on whether to send out neurotransmitters, and then it will later to inhibit or excite some later other neurons for passing on a message. Perhaps it is sensing, like sensing a feel or touch or movement. Our nervous system is one of the four main tissue types, the others being the muscle tissue, connective tissue, and epidermal tissues or epithelial tissues. So nervous system is quite the hefty part in our body. Now, we have the mechanism for the nervous system. It starts off at a negative 70 MV potential relative from inside to outside. So insides are more negative than outside. This is because we have potassium K as well as sodium. Potassium has equilibrium potential of about negative 90, whereas sodium has about positive 60. And the balance, the net equilibrium balance, gives about negative 70 equilibrium potential for resting. Now, it leans a bit more towards potassium because there are pores inside the membrane that slightly more leaky to potassium. So potassium ions naturally want to leak out, whereas sodium naturally want to leak in. So at rest, ATPase actively uses ATP to pump sodium out and potassium in to keep that gradient up because they naturally diffuse down the gradient. Alright, then in the process of depolarization, if incoming signal increases the membrane threshold or the potential up a little bit closer to negative 50, maybe you fire off and add some little bit, about negative 60, it's still not going to hit the threshold, so it's still not going to fire off until when you hit negative 50, you're going to set off the action potential and it's going to set off. Think about holding a rubber band, right? If you hold a rubber band like this, as long as it doesn't come off the tip, it's not going to shoot. So it'll either shoot or not shoot. But the moment it hit the threshold here, boom, it's going. So once it hit the depolarization, sodium gates open at the nodes of Rambier. Those are in between the myelin sheaths. And what happens then is sodium rushes in and push the information all through outside. Think about a series of townspeople trying to put out a fire. They grab one water bucket and just pass it to each other and pass the water all the way towards the fire and try to put it out. Right? It's a lot more efficient than having one person grab a bucket, grab some water, run all the way to the fire scene, pour it out, and do it again. No, people just stay in one place and pass it all the way down. Now, easy way to remember how the process of depolarization go is think about neurons being bananas. And because banana contains lots of K potassium, during depolarization, Na rushes in to make it a salty banana. So sodium rushes in, boom, inwards to make it salty. The gates open during the depolarization phase. Depolarization, think about polar as in like very negative apart, but as it gets closer to 0 and up to 35, all this is during action potential, but once it reaches there, the Na gate would start to inactivate the H gate would close. So what happens then is the sodium slowly loses concentration from the inside because it gets pumped out by the ATPase. But also, the mainly why it's dropping is because potassium is coming outside as well. Potassium is trying to get outside due to their negative 90 inert equilibrium potential. So they're climbing outside, and it overcompensates. It gets all the way down to negative 80, negative 90 MV, millivolts. So it reaches hyperpolarization. The potassium gates stay open until it reaches here, where it's so hyperpolarized that every gate closes, and for ATPase to slowly bring it back to the resting potential of negative 70. Alright, so how does the neurotransmitter work? Well, they are originally kept in the little vesicles in the synaptic terminal. Then, when information hits down here, it will then trigger calcium receptors to allow calcium binding, and it will trigger the release of the vesicles towards the membranes, and they'll get released outside and exocytose the neurotransmitters, which will then either bind to the next neuron or make them excited by binding onto the receptors, or inhibit them, such as GABA action. Right? The neurons, some neurons could also release neurotransmitters that self-activate more by having a transductory signal all the way back here to trigger positive feedback on itself. But typically, a negative feedback is in play because you don't want to constantly loop in an endless cycle, right? So we have certain types of other parts, not part of a neuron, but they're also supporting cells. Ependymal cells are for craniospinal fluid production. It's all the way in your third ventricle in your brain. Now, cerebrospinal fluid is very important at recycling nutrients, passing around, getting out wastes, production from metabolic wastes, and also cushioning against impacts against your head, also making sure that the fluidity, maintain fluidity between the parts of your neurons, just overall nourishment media. Schwann cells are the myelin sheath on the axon of the PNS, the peripheral nervous system. Oligodendrocytes are the same sheath but on the central nervous system for your brain and your spinal cord. So peripheral nervous system includes everything else other than your brain and spinal cord. The way I remember this is Schwann. Schwann kind of sounds like schlong, and schlong is your PNS, right? So easy to remember, Schwann, PNS. Astrocytes help nourish your neurons as well as maintain the blood-brain barrier. The blood-brain barrier keeps certain materials in while certain materials out. Common materials that go in are small steroids and small lipids, but glucose and bigger molecules cannot pass through, and proteins. They will need to be selectively permeable to it. Right? This is to prevent such as misfolded protein accessing inwards or certain things to clog in your brain. Now, microglia are, think of it as like the white blood cell around your nervous system. So around your brain and spinal cord, they will eat and gobble up any pathogens and keep you healthy. Now, last part, we have our spine. Right? Within the spinal cord, we have the dorsal side, which is towards our back, and ventral side in our front. The dorsal side receives sensory information and travel upwards towards the brain, whereas ventral side, it will take information from our brain and pass it down to the peripheral and pass it down to your sites and to activate muscles and parts. Now, the dorsal pathway takes in information from the peripheral outside and go up to the brain. Now, one thing is these are for somatic sections for the nervous system. Remember, there's also the autonomic part, which includes the parasympathetic and the sympathetic parts. Those would actually need to synapse at a ganglion. The ganglion is like another part, a neural structure that has postganglionic neurons that will then pass it on to the respective parts for your heart, your breathing apparatus, and digestion and such. The reason for that is to think about a parcel. You want to pass a parcel to a local mail carrier like Canada Post for them to do all the delivery for you. You don't. It's much more efficient, or not as efficient, but a little bit safer to keep it regulated. All right. So that's all from nervous system. Good luck.